All right. Carbon, of course, allows for count them, 4 covalent bonds. So the professor showed a train. That was to show long carbon chains. So the carbon chains are called hydrocarbons because these carbon chains, the hydrocarbons, have hydrogen attached to them. So hydrogen, having 1 electron only in its shell, readily makes a covalent bond with carbon, and they share those electrons. So methane is the simplest. Ethane, 2 carbons, propane 3, butane 4, ethene has 2, but they're double bonded, etc. So those are called hydrocarbons, and they're the basis of our macromolecules. Hydrocarbons don't just work alone. There's not usually just carbon and hydrogen. That would be a saturated situation. But often they have functional groups, and it's the functional groups that that function in our bodies for as hormones, for example, being able to bind to cells, and our ability to break down glucose is largely due to these functional groups, the hydroxyl groups. So as a result, glucose can be easily hydrolyzed. So these functional groups are groups of atoms that are attached to a carbon backbone, and they determine the properties of any of our organic molecules. And here are some of the functional groups. OH, hydroxyl group. Those are your alcohols. And alcohols are very volatile, so they easily dissolve and they easily burn off as gases. So alcohols are used as fuel, and they're often used as solvents. Ethanol is an alcohol that has 1 hydroxyl group. Carbonyl is another functional group attached to a carbon backbone, and they are your aldehydes, for example. Aldehydes can cause odors like cinnamon. Cinnamon has an aldehyde that causes that cinnamon odor. And it looks like this. An aldehyde has 1 carbon at the end of your chain of carbons, doubly bonded to an oxygen and singly to a hydrogen. The ketones are a little bit different in that the rest of the molecule is on either side of that central carbon, and it has a double bond to oxygen. Ketones. Carboxyl groups, they are your acids. So a carboxyl group is a carbon double bonded to oxygen, but singly bonded to an hydroxyl group, which the hydrogen of that group is readily given off. So that is what an acid is. It's a donator of hydrogen. So it readily gives off hydrogen. An amino group is another type of functional group. It has a nitrogen with 2 hydrogen atoms attached to the nitrogen. An amino group is basic. So that's the property of an amino group, like methylamine, for example. So these functional groups, have different properties. The phosphate is a functional group. We saw that already in ATP. It has 3 phosphate groups attached to it. So here you can see what it looks like with the central phosphate double bonded to 1 oxygen, singly to 3 other oxygens, which have negative charges. So it's a negatively charged group. The sulfhydryls are a sulfur atom and a hydrogen together as a functional group, and these provide very strong sulfhydryl bonds in between parts of a protein. So they make a protein structure very strong, very strong. So we use terms in chemistry called monomers and polymers. And monomers are the smaller single molecules like glucose, for example. An amino acid is a monomer. And polymers are larger groups. So the definition of a monomer would be a subunit. No, I won't put it over there, hang on. Monomers, a subunit of macromolecules. Proteins have 20 different monomers, 20 different amino acids. Polymers are simply a series of monomers bonded together. The term polymerization refers to the bonding of the monomers together. Polymerization. Yeah, so what this is showing you, this picture here is from what's known as gel electrophoresis. So in gel electrophoresis, you have a gel plate with agar in it, and you have some wells at the top. And then you put a product, some kind of chemical, this is how DNA is sequenced, for example, into the well. And then there's an electric current, generally from negative to positive. So something that's negatively charged, as DNA is, will travel in this direction. And the smaller particles, because they're smaller and they can sort of wind through the agar more easily, end up being further down the gel. And the larger particles tend to be up here. And that's how chemicals can be separated and identified often. So for example, these blue ones are longer and larger than the red ones. So if you know the size of the blue ones and where they end up on the agar gel, and you know the size of the red ones and where they end up on the agar gel, then if you have a mixture, as in here, you can see that the bands line up, and you must have both of them present. So in this case, so these are all monomers. In this case, we have the possibility of dimers. Dimers are just simply two monomers together, di, two. Yeah, so in this case, we have dimers forming here, and we've got these dimers forming as well. But here we have an extra kind of dimer, which happens to be these molecules together. So now we know that the blue monomer can bind with the red monomer in this case. So it's just a method of finding out what kinds of molecules we have. So this method, it's called gel electrophoresis. And it's a very effective method for, in forensics, for example, of figuring out the parentage of an individual, or the blood from a suspect, for example. So when we bind those monomers together, we use a method known as dehydration synthesis. It's also known as condensation. It depends which textbook you look at, actually. So yeah, monomers bond together to form a polymer. This is known as synthesis, and it happens because a water molecule is removed. So that's why it got the name dehydration synthesis or condensation. So the water molecule is removed. In other words, the hydrogen from one monomer binds with the hydroxyl group from another monomer, thus making a water molecule. And the two monomers are covalently bonded together now. That's dehydration synthesis. Hydrolysis is splitting a polymer. Lysis means to split. And you add a water molecule. Yeah, hydrolysis. Hydro stands for water, lysis stands for splitting, hydrolysis, splitting using water. So when you add a water molecule, the one hydrogen group ends up on one monomer and the other hydroxyl ends up on the other monomer. And that's how splitting happens. That's how digestion happens. So let's move to organic compounds in a moment. I'm going to stop the lecture now